So this is Singapore, a fairly wealthy nation in Asia. And like many countries in Asia, it's plagued with dengue fever. In 2022 alone, there was nearly 50,000 cases of dengue. So how exactly did the officials get rid of the mosquitoes transmitting the virus? They released 24 million more mosquitoes into the wild. Yeah, seems a bit counterintuitive, don't you guys think? Well, in reality, each of these mosquitoes are genetically modified with a bacteria that makes them susceptible and unable to transmit dengue to humans. This bacteria being wall bacteria. These 24 million mosquitoes would then go on to mate with a larger mosquito population. The gene would propagate, and thus this would neutralize dengue altogether. Now, it's important to note that as genes propagate inside of a population, their effectiveness gets diminished and essentially becomes diluted. So obviously the Singaporean officials don't want to create 24 million mosquitoes every year. That doesn't seem like a very fun job to do. So what do they do to get around this? They use a novel technology known as gene drives, which ensure that the gene gets passed on 100% of the time. This is in contrast to traditional Mendelian inheritance, where the gene has a roughly 25, 50, 75% chance of getting passed on every single time. So how exactly do gene drives work? It's a very interesting technology if you look at it. There's two copies of every gene that we get, one from mom and one from dad. Let's say that we've genetically modified dad's copy to have the gene drive. And I want you guys to all think of it like a virus of sorts. It's going to hijack mom's copy and overwrite the genetic information from mom's copy to dad's copy, thus creating two copies of the same gene. This way, regardless of which one the offspring inherits, it's going to contain a copy of the gene. So this is all cool and well, but this all got me thinking, how exactly can I apply this technology to maybe another virus instead of dengue? I mean, as Singapore did dengue, they were propagating a gene in the population. But let's say I wanted to remove some virus inside the human body. Then I would need to propagate a gene inside the human genome. And this is when I found HIV, which is a devastating disease that I'm sure you guys all know about, but for some of you that might not, it's a bloodborne virus that attacks the body's immune system, rendering it useless. People that have HIV are predicted to have less than 10 years of life to live. And this is tragic. And that's why, and current solutions and treatments for HIV are pretty disappointing as well. I mean, the closest thing we have to a cure simply limits the spread of HIV. And this in itself has a wide variety of harmful side effects. So I would hardly consider it a cure. And that's why I'm introducing HIVax, which is a novel HIV treatment that's able to cure HIV and stop it from growing within the body. So how exactly does HIVax work? It involves a spacer DNA, and I want you guys to all think of the spacer DNA like a sleeper agent of sorts. It's hibernating inside the cell until it gets activated, the activation mechanism being the presence of HIV inside the body. Once activated, it initiates several components, notably being the HIV-1RT, which is the actual detection mechanism for HIV. There's also the Landor repressor, which initiates the apoptotic pathway, otherwise known as cell suicide. And lastly, we have the self-cloning CRISPR, which is a necessary component in all gene drives. So, now that you guys know how HIVax works from a holistic view, let's look at the three phases. The first phase is the delivery phase. How exactly do we take our gene and put it inside the cell? The second phase is the elimination phase. Once the cell is inside once the gene is inside the cell, what exactly do we do to detect the HIV and how do we eliminate it? And lastly, the propagation phase. How exactly do we get our gene to propagate inside the genome and thus build immunity? Firstly, the delivery phase. So the way we're going to be delivering this is via lipid nanoparticles, which are these small little balls of fat that have been popularized after their usage in the COVID-19 mRNA vaccine, which I'm sure you guys have all heard of. These lipid nanoparticles are so useful in taking genetic information and inserting it inside the cell's nucleus. Once the gene is inside the cell's nucleus, it's creating these HIV-RT transcripts, these pieces of RNA, and they're just floating about in the cytoplasm. And these are keys of sorts. These keys obviously are specific to a lock, the lock being the HIV RNA. Once the key meets its lock, it's going to bind to it and close like a zipper, releasing the Bax protein, which initiates apoptosis. Now, just before a cell sacrifices itself and grow a death to preserve the other cells in the body, it's going to create hundreds of copies of our gene and coat them in these lipid nanoparticles, thus continuing the cycle and propagating the gene further. It's also important to note that if our cell, by any chance, doesn't come in contact with HIV in its lifetime, it's just going to continue to live a healthy, normal cell life and multiply via mitosis, again, passively spreading the gene. It's possible that in several years, HIVax will become the benchmark treatment for HIV, and it's going to become another one of those standard vaccines that you take as a child. I'm Christian Mandapara, a 16-year-old innovator at TCAS, and this was HIVax. Thank you.